The same way that we cut open the brain?
Pretty much. Yep. So
Okay.
where you're making kind of single incisions essentially the whole way. All right. So we're essentially going to kind of pick a line where we're connecting those two things and we can see our ventricles here. So what we're going to do is you just want to kind of score it.
I can't see.
Okay.
Showing
So
me
that
the
you can kind
tray.
of see where your line is going to be. Okay. And just kind of work your way around. Now, I will say that this is not the best tool for doing a big chunk of meat like this. Okay. We would rather have like a big chef knife or something like that. But for our purposes, this will still work. So just now that we have a line, now you just want to kind of start to slowly work at it and go deeper and deeper each time around. Okay. If you run into something too dense, you might have to stop a little bit. But our goal is not going to be to stop and saw. Okay. It's when you stop and saw that you're going to kind of tear stuff up. And that's really not what we want. Okay. So just like I said, kind of just keep working your way around. You can also just kind of start on one side and then just really make that one side cut, get it opened up. As you cut it more, you'll be able to kind of put some pressure on it to open it up even more so you can cut a little deeper and so on and so on and so on. Okay.
What happens to the blood and stuff? Do they like
They've all been drained. So whenever animals are harvested either for this or for meat and stuff like that, they will drain them of as much blood as possible.
Yeah.
So that we don't have to deal with all those liquids and stuff like that. And then in the case of something like this, it's then going to be injected with a fixative.
That's cool.
And that fixative is what's going to kind of harden these a little bit. And that's also kind of what you're smelling. Those fixatives are also going to be dissolved in alcohol. So that's a big part of the reason why whenever you've done a dissection, the smell tends to be very noticeable.
Yeah.
And that's just because it's alcohol stuff. It tends to be very aromatic. Like it tends to dissolve into the air very quickly. Or evaporate very quickly.
Man, my hands are so shaky. I'm not gonna do surgery. I'm not made for that.
Well, doing open heart surgery, you wouldn't be doing it this way anyway. Be a little bit more to it. No, you're doing great, actually. That line's pretty straight down in there. Trying to keep it where she can see it as well. It's having tough there.
Yeah. Especially on the top. It's a little bit tougher.
Hear that?
Yeah, I like the sound of that.
That is a valve.
Is it because it's tougher?
Here. All right, I'm gonna kind of hold this in place. All right, don't cut me, but take the scalpel and you're gonna kind of start it kind of roughly right here, just under, just near my finger, and work it towards you nice and slow. And you should be able to feel that valve.
Am I even?
Yep, just put a little bit of pressure on the end of it.
Oh.
So that's muscle. That is the valve. Did you feel it get hard?
Yeah, like it's like a
Yeah, do the same thing. And you can see it even down in there, the white part.
Oh, I do.
Bring it back over here. Let her do a little bit more. Here, you can go ahead and open us up this way a little bit. Let's see how different the meat feels versus the valves.
Yep.
Yeah.
All right, you're getting into another valve there. See those little chordae that you're cutting.
Those. Yeah. Wow, you can
Keep going. Hold on.
actually
Keep going.
see-
Keep going.
Hey.
Oh.
Well done.
Wow.
I mean, you want to keep where you have what you have. You
It's
can.
like a little hot dog bun.
Pretty much. All right, and what you just cut was the serous membrane.
Yeah.
That little pop that you just heard.
Oh, isn't that it right there?
Mhm. And I'm sorry, Jennifer, I really am doing the best that I can. Unfortunately, like we're too far away from the desk for me to be able to see it on the screen. So I'm having to like bend down while we're seeing it from here. All right, so yeah, now we have the inside of our heart. Y'all did good. It's not too torn up at all. But yeah, there's one of those valves. Oh, we can actually see an entire cusp of this valve. So grab that. Hear that?
Mm-hmm.
Grab it. You can't break it.
What the heck?
Like it's dense connective tissue. So it is literally dense. Okay. We can also see our blood vessels up here. Like we can see thick walls of that guy right there. Like that tells us that's going to be really springy and really tough. Now we can also see the differences. We've got two ventricles right here. We have this one where my index finger is, and then we've got this one over here where my thumb is. Which one has thicker walls? This one or this one?
This one.
Yeah.
This one by a lot. Like
Yeah.
last time when we saw those pictures on the screen, it was like, oh no, he's probably like they're a little bit different, but they're not that different. No, they are massively different. Okay. Why? Because this has to push to your entire
Mhm.
body. So all the way down to your little toe and everywhere else in between. This just has to push the lung. So this
Mhm.
one's very, very thin by comparison. Still fun though. We've got our valves over here. We can see our papillary muscles attached to those little cords. So we can see those nice little cords there. Just tore one. That is our chordae tendineae. Okay, so chordae tendineae, and remember it's connected to these little bands of muscle called papillary muscle. And remember, these are used to open the valves. Okay, they're also connected to these muscles so that the valves can't go up,
Mhm.
so that the valves can't extend all the way into the atria, because that would be bad. Okay, we need those things to be separate chambers. Let's see here. Did we get lucky? Wait, we did. All right. So see a little hole right there?
Uh-huh.
Okay. So I'm gonna point it down here. I can't tell if it's on here now. So right there where my little pointer is, that is going to lead
Yeah.
into our coronary sinus. That's what all this stuff is back here, is our coronary sinus. And that's where that little hole leads. It's because that's the entrance of our coronary sulcus.
Mhm.
Okay, or coronary sinus. So that's basically where all that blood that feeds all this muscle gets returned to the heart. So that's that little kind of shunt. Let's see. Oh yeah. So you actually cut right into the opening of the aorta. That's what this is right here. So you can actually see the pulmonary semilunar valve. So that's what that is. See how it's that kind of half moon shape even. So you can see that shape of that cusp. But that is our aorta.
Huh.
So that means that is our aorta. And when you grab it, it's tough and springy and strong. Why so tough and springy and strong? Because it's subject to massive amounts of force. Okay. So yeah, y'all can kind of keep exploring these things. The other little ridges we can see in here. Oh, that looks like it was cut. Interesting. Some of the other little ridges in here, like those little bumps and stuff like that. These are basically all extensions that allow the heart to stretch as it's filled with more and more blood. So we call those the trabeculae carnae. Remember that trabecula means bridge. So it's basically bridges of meat. They're literally bridges of meat. Okay. So that's what those little ridges are and stuff like that. So it's quite fun. So yeah. You want to kind of keep exploring. And if you wanted to make any other cuts because there's something you wanted to see that you can't really see in this particular angle and you want to cut something else off, that's totally fine. Like I said, this is for y'all.
So cool. This one's bicuspid, right? Because there's two.
Yep.
That's so cool.
Actually, no, sorry. This is on that side. So this is the
So
small
it's
side.
this way.
So yeah. So this is
Okay,
the
so
tricuspid.
tricuspid.
Yeah.
Okay.
Yeah. So the bicuspid's on the thick side.
Yeah.
But you can see just how big and thick those things are. Like, it's just crazy.
Ew. Woo. What's the like? Is that the stretchy part? Does it? Yeah.
Yep.
It has interesting like texture.
It does. It does. Once again, that's so that we can fill it even more. So that we can put more blood into it so that when the heart beats, we can push out more blood with each heartbeat, which is what happens during things like exercise.
And like blood pressure and stuff.
Mhm. Like I see you grabbing, like it's tough.
Yeah, I know. It's
Now you know why I told you that story about that my friend like crimped
Yeah,
himself? Like,
yeah.
you probably thought I was joking. Like, okay, you couldn't actually hear. It's like, no, you could literally hear him. At some point, I will sit down and like make one of these like with a good camera, so I can sit down.
But- Like I can just imagine it.
Mhm.
But how can it get so like? Because it's so tough. Like how does it?
Well, so part of the reason it's so tough right now is because of the preservatives. The
Mhm.
preservatives, whenever they hit meat like this or like muscle, it does tend to make it harder.
Mhm.
Like real ones are firm, but when they're firm, when I say they're like, and they're more firm even than something like with skeletal muscle.
Yeah.
Just because cardiac muscle tends to pack itself more densely.
Mhm.
Because keep in mind that with skeletal muscle, every single muscle fiber has a layer of connective tissue around the outside of it, right? So there's a little bit of space in between those cells. So anytime you've held like meat or something like that, there's some gapping
Mhm.
in between each thing. And then we've got bundles that have their own little gaps and stuff like
Yeah.
that. We don't really have that sort of structure in the heart, so it tends to be more dense. And all we've done by preserving it is, or all we've done when we preserved it is we've made it more dense and
Yeah.
harder. But if you find one that is raw, like you can actually go to the butcher shop and buy a raw heart. And like it is softer than this, but it is still harder than something like a steak
Yeah.
or harder even than something like a brisket. So I'm trying to think of like the harder meats that I've held. And it's still significantly firmer than that.
Mhm.
So it's not as hard as it is right here. Like you can still
Yeah.
pull on it. But it is still firmer than like what you would find in even really tough meats, like a
Mhm.
brisket or a skirt steak or something like that. It's still tougher than that or harder than that.
That's so cool.
It's pretty fun.
Mhm.
Like they're fun to just kind of play with and like see what you can find.
Yeah.
You want to see this?
Sure. Oh. Cool. Oh, okay. Because you can see like the valve really good on the. You can see the valve really good on the.
Yeah, you can see the valve really well now. You can't really see much of what the atria in this case looks like, but you can see the valve really well.
see in there.
Yeah, you can see in there. But I mean, if you want to section something out so you can see it, that's totally fine. Yeah. Here. You want to section something out?
Thank you.
Seven.
Ooh.
Oh yeah. Okay. Yeah. No, you did good. You didn't move much, but you may have read some. That was good. That was a good cut.
Thank you.
And this might even be a fun time to take one of those probes and see if you can find one of those pulmonary arteries. There you go.
Right through there.
So, no. So that's just one of the little pockets of the world. Okay.
That's so cool. Like, see that? That's the little branches. Like if you open it up.
Yep. Trabeculae carne. But this was also why we looked at the anatomy and stuff last time, was so that you are pretty comfortable with and had an idea of what you're going to be looking for before we did this today. Jennifer, I do think there are some virtual labs to at least kind of be working on and stuff right now too. I don't know if I don't think there's a dissection one, but I do think there's a cardiac physiology sort of overview kind of lab. Hmm? Oh yeah, now you can see in there.
And then which part is that just one of like the
That's probably just a slice of the aorta.
Mm.
It's super fun. Y'all have a good look at yeah, you still have some nature over here to kind of see on that side too. Yeah.
That's so cool.
Sometimes you can even find the fossa vallis as well.
Ooh. Oh my goodness.
Yeah,
Look,
see?
you can see my finger.
Yep. So that's in the coronary sinus. But yeah, it's so thin that you can see through it. But yeah, that's the coronary sinus.
Yo.
So see how it's between. So like we've got all these little grooves on the surface, right? These sulcuses, right? And that's where those blood vessels live. So when you did that, you found that big one, that big sulcus in between the atria.
Yeah.
So that's where that line is. See? Exactly where you expect those big blood vessels to be.
Uh-huh. That's good. That's so cool. Whoa. That's so cool. And these are just the same thing but tiny.
Yep. Smaller, probably tastier big.
Tastier.
Hmm?
Did you say tastier?
Mhm.
The smaller they are, the tastier they are?
Well, not necessarily. Smaller animals tend to be more mild in flavor because they've had less time to accrue a bunch of like weed more pungent sort of flavor chemicals.
Yeah.
From their foods and stuff like that. So they tend to be a little bit more mild.
Mm-hmm.
So like veal parmesan, like say you've had veal or something like that.
Mhm.
Veal is a less potent sort of beef flavor.
Yeah.
Than from an older cow.
Mm-hmm. That's cool.
But like typically like what you eat when you eat beef from a restaurant or you buy beef at the grocery store and stuff like that, that is from an animal that is around a year old, like between 9 months and a year old. Either a heifer or a steer.
Mm-hmm.
Usually. Now, if you get meat from, say, a family farm, you can actually sometimes get meat from older animals that have had a chance to go through birth cycles and stuff like that, or older, and that meat will be more flavorful.
Mhm.
Like there will be more color in the meat, there will also be more flavor in each bite. However, it will also have accrued more connective tissue,
So
and it will be tougher.
Tougher, yeah.
So it will have more flavor, but be tougher, which means that kind of stuff has to be cooked longer.
Mhm.
And we see the similar stuff when it comes to like chicken. So when you buy a chicken at the grocery store, like chicken breast or something, that is from an animal that's like 6 weeks old, like 6 or 8 weeks. They're young. Okay.
Yeah.
And that's great for like fast cooks and stuff like that because they're going to stay tender and all those kinds of things. But that's not going to get you the most chickeny flavor of chicken.
Yeah.
Okay. So that's not the best stuff for like making a soup. If you're going to make a soup, you usually want something that's older. And there will be like different words and stuff that you'll find like on chickens made for like stewing and stuff like that.
That's cool. Is that why like people age their meat?
No. You age meats because you essentially are able to get a lot of the like particularly like dry aging meat, which is usually how people age it as far as like, oh, it's a 30-day age dry aged steak, like this big steakhouse. So they do that to pull as much water out as they possibly can to essentially concentrate the flavor in it.
Mhm.
And also they will grow a little bit of mold. And it's actually like part of the aged flavor that comes in there is actually from some of the mold kind of working its way in. That's a little bit of the sort of aged flavor that comes into those meat, but it's also like it's mainly there to concentrate it, like getting out a bunch of the water so that you're left truly just like with the myoglobin and stuff like that that's in the meats. So it's a beefier tasting beef kind of thing.
That's cool.
Yeah. But that's also why that stuff is expensive because you have to have a place to let that stuff sit for 30 days and take care of it and all that kind of stuff. And so you're not only going to lose weight in water, you'll lose a lot of weight in water, but you'll also lose a little bit of weight in meat that you might have to trim off because of gross looking molds and stuff like that.
Yeah. Do we put these in that red one?
Yes, the
Okay.
gloves and stuff need to go up there. Are you good? Take all the pictures you need. Did you want to take any pictures? Okay.
My mom thinks this stuff is cool.
And then, all right, Paul. Super fun though.
Super fun.
There's only 2, so I don't mind cleaning it myself. All right, y'all need to go wash your hands. Whenever you're done washing your hands, I will have the table cleaned, and then we can start talking about EKGs and stuff. Don't worry, we're not doing the hard part. Yeah, there will be some lecture and stuff. We're going to talk about the beating of the heart here in a little bit, and then I'm going to have them do some laps. There's brain, or not brain, there's heart on my computer. It's fine. It'll be okay. I have no, like, I would, like, you shouldn't, but like, I would feel way less bad about grabbing one of those, like, with my bare hand than I would, like, if we had a saliva lab. When we do the digestive lab, like, I have, at one of my old schools, we did a lab where you would, like, you'd have everybody in class spit into a cup for a while and collect like 50 milliliters of saliva from everybody. And that was the most disgusting thing in the world. God, it smelled so bad. Huh. It was horrendous. I refuse. Like, we will never do that in here. Like, no, like, I don't care. Like, I'll eat that before I would have a saliva lab. Like, no. Like, I would rather do that than I have another saliva lab. It was horrendous. Ugh.
Where they all spit into a bottle before they live to lower their weight.
What? That's disgusting.
I know a ton of people that were like real close to like this weight class and they wanted to get to a lower one, so they just spit in bottles on it. It's so nasty.
Yeah, nope, 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 nope. Not worth it. All right, so what are we doing here? Don't need that screen. I need this one. Wait, don't need that one yet. All right, so now what we're going to do is we are going to talk about how we get the heart to beat. Now, before we do that, we do need to understand a couple of things. Namely, that the heart is composed of cardiac muscle cells, and the cardiac muscle cells are very, very different than skeletal muscle cells. One of the big differences between cardiac muscle cells and skeletal muscle cells is, of course, going to be this shape. Okay, cardiac muscle cells have branches very often. Okay, this means that one cell is sometimes connected to as many as 3 or 4 other cells. Okay, now that means when you generate an action potential in the membrane of this cell, well, what's going to happen is that it's going to generate an action potential in the cells connected to it. So by having all of our cardiac muscle cells connected basically in this big network, when one cell generates an action potential or one group of cells generates an action potential, we can then propagate that action potential to all of the other cardiac muscle cells. That's great because now one set of cells can tell the rest of the heart what to do. Okay, that means we essentially have one site or as many as one side of control if we need it. Now, one of the other ways that our cells are able to talk to one another is because in addition to those branching points and just their sarcolemmas touching or their membranes touching, we also have the cells connected via what we call an intercalated disc. Okay, so these intercalated discs are essentially the connection points between individual cardiac muscle cells. Now, where they're touching, they're not just kind of butted up against one another and welded there. Instead, they are held together with a bunch of little rivets. Okay, so a rivet has a hole in it, importantly. Okay, so that little hole in between our cardiac muscle cells, we're going to call gap junctions. But these are little holes, which means if we change the chemical composition of this cell and it's touching another one and it's open via that connection, well, what's going to happen is it's going to change the chemical composition of this cell beside it, right? So we now essentially have intercell communication because of these intercalated discs and because of these little gap junction proteins that we see down here. Does that make sense? Now, to help us get more surface area to have more of these things, we're also going to have this kind of interlacing pattern at the junction between these cells, okay? Now, one big thing to keep in mind with our cardiac muscle cells, though, is that unlike our skeletal muscle cells, where our skeletal muscle cells can utilize anaerobic respiration for a while, like they can work without additional oxygen getting fed into them, we don't have that ability in the heart. We need oxygen essentially all the time. Now, one moment here or there without oxygen isn't a huge deal, but we can't last multiple seconds without oxygen in these tissues. So that means we have to have a massive amount of blood going to them all the time, okay? If oxygen runs low and any bits of these tissues go ischemic, those cells can die. Now, can you think of any scenarios that might lead to some of these tissues going ischemic? That's to say that they are running low on oxygen for any period of time. Being underwater, yeah, that might be a problem because at that point, the entire body is going to go low on oxygen, right? But what about if we just isolate it just to the heart? Can you think of anything that might lead to just the heart or even just portions of the heart getting low on oxygen?
Like what?
A clot, exactly. An MI, a myocardial infarction, a heart attack. Heart attacks are caused by a clot. They're caused by clots in those coronary blood vessels, in those blood vessels of the heart. If you block blood flow to any of those portions, all of a sudden, all of those downstream portions aren't getting blood, and those tissues can go ischemic and die. Now, if it happens and we're a branch of a branch of a branch, and we lose a handful of cells, that is a minor heart attack. Those cells can die. As those cells die, they're going to release some particular chemical components into the blood that says, hey, these are the remnants of cardiac muscle cells. You've had a myocardial infarction, you've had a heart attack, okay? If it happens in those first couple of branches onto the left or to the right side that have branches coming off of those, all of a sudden you're shutting down blood supply to basically an entire side of the heart. What do you think happens then? It is a heart attack. It is an MI still, but now it is a major heart attack. Sometimes those major heart attacks are so bad that the patient wouldn't be alive to get out that door. They would fall down and they would be dead right there. And there's not a whole lot of intervention that is possible to have been done in any real moment to have saved them. The blockage was so bad in particular places that they can just sometimes die. That is why that right coronary artery is oftentimes called the widowmaker, because if you have if it blocks there, well, you just shut down blood flow to the entire right-hand side of the heart and nothing else matters. Like there's really nothing you could have done. So those are very, 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 very bad. So we want to avoid that. So that means we want to avoid those clots and stuff as best as we can. But aerobic metabolism is required here, okay? So just kind of keep that in mind. So we have a massive amount of energy demand going to places like the heart, okay? Also fun, the heart can use any number of different types of fuel molecules in order to work. Fatty acids, still fine. So your heart cells can do fatty acid metabolism. They can do glucose metabolism. So that means they can do sugar metabolism. They can do lactic acid metabolism. That means they're literally able to break down lactic acid and turn it into ATP. Amino acids, so proteins. So that means they're able to break down proteins and turn those into ATP. Even ketone bodies. So that means even if your body is under ketogenic acidosis, so say you're under like a diabetic that isn't able to pull glucose out of their cells and stuff like that, so you produce a bunch of ketones as a byproduct. And you have a bunch of these ketones floating around in your blood, your heart is even able to break those things down. That basically tells you that your heart is very, very efficient at getting energy out of anything and everything that it can. That is different than something like your brain. Your brain works off of glucose. That's it. Full stop. Okay? Which means we have to maintain some amount of glucose in our blood. Otherwise, our brain doesn't work. Okay? However, keep in mind that your body can turn lots of all of these other things. So it has the ability to turn fatty acids into glucose in the blood. It has the ability to turn literally anything on this list into glucose in the blood. if necessary. It just doesn't like to all the time. That's also part of why your body is like, no, I want sugar and I want sugar now, because it's the easiest thing and it's the thing that our brain craves the most. You could even make the argument that we see the color red because of sugar, because it allows us to distinguish ripe fruit from unripe fruit. So that means by seeing the color red, we know that, hey, that fruit will have a higher percentage of accessible sugar in it so that we can eat it. Now, getting back to the heart. So in order to make the heart beat, the heart has what we call a conduction system. This basically is going to be a pathway within the heart that allows us to transmit action potentials from one cell to the next, okay? And a particular pattern that those action potentials are going to go, okay? Now, importantly, these are not nerve cells. These are cardiac muscle cells. Many of these cardiac muscle cells do not even have contractile fibers in them. No actin, no myosin. Most of them have some, they're just a very small amount. So their main job is not going to be contracting like most of the other muscles will. Instead, it will basically be to act as a conduit for an electrical signal. Okay. Now, the reason it's going to be important, though, is because we have one set of cells that gets to determine the speed of our heart, and that is these guys right here. This is what we call the sinoatrial node. This is also called the pacemaker of your heart. Okay. So the sinoatrial node here is a collection of what we call nodal cells. So these are cells that are autorhythmic. They will beat at a particular pace, or they will start an action potential at a very particular pace. In this case, it's going to be between 80 and 90 beats a minute. Okay. So without any sort of external input to the heart is going to beat 80 or 90 beats a minute because this guy said so. Okay. Now, the way that's going to work is that we have a bunch of leaky channels in here. So usually our voltage gated channels and stuff like that open only in response to particular changes in charge. In the nodal cells, some of those voltage gated channels are leaky. The valves just don't close all the way, and they're going to allow some amount of chemical to pass through, which means even after we have depolarized and repolarized, we're going to see this slow creep up, or this slow change in membrane potential until we reach the threshold value that tells us to trigger the response. Okay? So that's going to be very different than what we saw in other muscles and also what we saw in nerve cells. Okay? Where whenever it changed, it would basically wait until it received a signal to change again. This time it changes, and then it will start the process, and it will get closer and closer and closer to its threshold potential before it does it again. Okay? And that's going to be decided here at our pacemaker node. Okay? Now, after that signal leaves our pacemaker node, it's going to then travel to all of the cardiac muscle cells that are attached to it. So that means it's going to move around basically the entire atria, both the left and right-hand side of the heart. Okay. Now that signal will then travel down and reach a special set of nodal cells down here in between the atria and the ventricles. This is called the atrioventricular node, or the AV node. Now, the AV node is special because this is a special set of nodal cells that is connected to this little pathway right here. Okay. Now, the reason that we need this to happen, so say we just followed that electrical charge from the SA node out and it hit everything in order. Would the heart beat the right way? Because if that happens, so we're going to contract the atria from top to bottom here, right? That's fine because squeezing from the top down, or like in, that's totally reasonable. However, when we get to the ventricles, all of a sudden we're going to be squeezing the ventricles at the top, which is also where the blood leaves the ventricles. So if we squeeze the top of the ventricles, we're also inhibiting the ability of us to get the rest of the blood out. We are squeezing the toothpaste tube in the middle, or even at the top. Is that ideal if you need to get the toothpaste out? No. Okay, so that means we need to figure out how to get signal from up here where it starts to down here to the apex of the heart. This is where the AV node comes into play. So connected to the atrioventricular node is this bundle of specialized muscle fibers called the atrioventricular bundle. That will then lead into the right and left bundle branches. Those right and left bundle branches are then going to break into the Purkinje fibers, which are what are actually going to tell those muscle cells what to do. Which means, notice that these Purkinje fibers are shorter than these. Why? Or that means it's an electrical signal. So which one does it hit first? Here at the apex of the heart. And then it goes up here inside kind of the lateral walls of the heart. So that those lateral walls of the heart will contract after the apex of the heart, which means we start pushing the toothpaste from the bottom. Okay, so this is going to cause the heart to shorten, or the ventricles to shorten. Why? So that we can drain or push out all of that blood from the ventricles and out through the aorta or the pulmonary trunk. Does that make sense? Everybody kind of see that that's what we're trying to do? Okay, so let's follow that signal again. SA node tells the atria what to do. Signal gets down to the atrioventricular node, which then goes down through the atrioventricular bundle, then finally through the right and left bundle branches, and then out to the Purkinje fibers, and that's what's going to tell the apex of the heart to beat. Now, one of the other reasons that the atrioventricular node is special is that it's also a set of nodal cells, which means we have a set of autorhythmic cells. These cells are going to tell the heart to beat around 50 to 60 beats a minute, which is something that you could survive. Okay, that's enough to keep your brain functioning and enough to live. That is not enough to do really anything else. Okay, so if we lose the signal from our SA node, the rest of the heart is going to be getting this big, strong signal from the atrioventricular node to beat at this much slower pace, which is enough to sustain life, but not necessarily enough to do anything beyond that. Okay, so that's something that we want to fix, but you won't die. Okay, now the rest of the heart cells are also autorhythmic, but they're going to be autorhythmic at around 30 to 50 beats a minute, which is not survivable, which means if we need or want the heart to beat fast enough for us to be able to live, we have to be getting that signal from either the atrioventricular node or the SA node. However, the SA node does things faster. Because the SA node does things faster, guess what that means? Which one are we going to follow? The one that's saying go this fast, or the one that's saying go, I can't even do it. So this fast, or the one that's going much slower? Well, if this one's saying beats and it sends out a signal to everybody, it just overrides whatever this other one is doing. So this is what we're going to do because the SA node says to do it. Okay. So now that means if we want to tell the heart to go faster, like say we're exercising and we want that, like that 80 beats a minute, 90 beats a minute isn't fast enough because we're doing something dumb, like jumping jacks, right? 90 beats a minute is not enough to sustain my jumping jacks for very long. So I have to speed up. So what tells us to speed up? The sinoatrial node, exactly. So that means we have to tell the sinoatrial node to go faster. What do you think we're going to use to tell the sinoatrial node to go faster? Where did we find the cardiac center in the brain? Makes alligators angry. For those of you that know really old movies.
Oh, no, not the medulla oblongata.
Medulla oblongata. Okay. It is the medulla oblongata that is going to contain the cardiac center. It has two major pathways. We have an acceleratory pathway that tells us to go faster. And importantly, we have a cardioinhibitory pathway that tells us to go slower. Okay. How is it doing this? Well, or where is it getting these signals? It's getting these signals from two major places. It's getting them from baroreceptors, which is to say pressure receptors in places like our organs and in our blood vessels, as well as chemical receptors or chemoreceptors in places like our blood vessels and our heart. Okay. Why? We're monitoring pressure. We're monitoring chemical composition. If O2 levels get too low or CO2 levels get too high, well, what does that mean? That means we need to beat faster so we can get more blood into the lungs and get oxygen in and CO2 out. Or if oxygen levels are running low in our muscles, we need to get more oxygen to those tissues, which means we have to beat faster. Does that make sense? Now, the same thing is, I guess, gotta love Adam Sandler movies is one way to putting it. There were some good ones a long time ago. Like Happy Gilmore and Waterboy are about the only ones that I think I could still watch. I had some friends that watched the new Happy Gilmore and they said it was not good. Really? That's good. Maybe they just weren't in the mood, I guess. I think you have to be in a certain mood for watching Adam Sandler movie. Yes. I have laughed very hard at Waterboy though. I also use that reference. I've used that reference for a very long time. Let's put it that way. Yep. Now, for these things, like we also need to be able to slow stuff down. So whenever we're not exercising, say you've just had a big meal. What do you like to do after you eat a big meal? Take a nap. You like to sit down, sit still, and relax. That rest and digest function is also going to come from the medulla oblongata. The medulla oblongata in this case is going to send a message through the vagus nerve. So that's this guy right here. Okay. So it's going to send a message down to the vagus nerve, down to the sinoatrial node that says, hey, sinoatrial node, keep it down. And it's just going to slow down the rate that the leak channels in the sinoatrial node open, which means we're going to slow down the pace from the sinoatrial node. Okay. And as long as that pace is above 50 beats a minute or so, it will still be enough to completely override any signal from the atrioventricular node. Okay. So that's what's going to make that SA node the primary kind of pacemaker of our heart. So cardioinhibitory center can slow it down via parasympathetic innervation and via the vagus nerve, or it can speed it up if we need to. But once again, it's all going to be because of signals that we're getting from pressure receptors and chemical receptors throughout the body, whether it's in our vessels, in our heart, or in other places of the body. Does that make sense? So let's kind of take a look at those. So here's, once again, these nodal cells. They have these special leak channels out here. So these are going to be open more often than they typically would be. And that is, they're going to slowly open until we reach this kind of threshold. Once we reach the threshold, that is when we initiate the action potential that then will signal all of the other cells. The rest of this is the same thing as what's happened in a nerve or a cardiac muscle cell. So there we have the inherent SA node rate. Oh, it's saying 100 beats a minute, so I was a little off. Okay, so we have the threshold met, and then we depolarize, we repolarize, and then we have our resting membrane potential down here. So in a normal muscle cell, this would basically just be flat until we're waiting to receive another signal, right? And so these channels are all closed. But some of these channels open at a particular rate, so it's going to have this slow increase in membrane potential until we reach the threshold value, at which point it goes off again. Okay, so we're going to see this kind of slow creep up, and then threshold, bam. Getting closer to threshold, bam. Get close to threshold, bam. Okay, instead of waiting for a signal that causes, and that's what causes us to go above the threshold. Instead, we're generating our own signal. We're generating our own change. Okay, so that's what makes these cells autorhythmic. It's basic, and all of that is because our channels are a little bit leaky. We'll see those here in a second. Now, like I said, SA node gets a little bit leaky, leaks out, and then that signal leaves, and then the signal is going to move from the sinoatrial node throughout the atria, telling the atria to contract. Atria contract, and then the signal moves down, finally gets to the atrioventricular node. Now, by doing this, one, this delays us contracting at the top of the atria, but this is also going to allow us to delay the signal long enough to make sure that the atria are emptied before we contract the ventricles, okay? So signal goes down to the AV node, which then goes down to the atrioventricular bundle, and then out through the bundle branches, and then finally out to the Purkinje fibers. Now, once it gets out to the Purkinje fibers, it then leaves these special channels and goes out into the actual contractile cells, and then we contract the heart. But this way we can start the contraction of the heart here at the apex and then cause the heart to shorten when it contracts. That way we can push all of that blood out, okay? So by having this little bit of a delay as we go from the atrioventricular bundle down to the Purkinje fibers, this gives us enough time to have the atria completely empty and then make sure that we can get the signal down here to the apex of the heart so that we're once again not squeezing the toothpaste tube from the middle. Okay, so we can start that stimulation down here at the apex and then eventually work our way up towards the aortic trunk or the pulmonary trunk. Purkinje fibers are also quite big, okay? We couldn't see them with these, but if you had any sort of a magnification, you could actually see that those cells themselves are actually quite a bit larger than the other cardiac muscle cells, which means we're going to get signals moving through them very, very quickly. That's also how we're able to get those sections to start to fire. Now, someone might need to have a pacemaker installed if for some reason parts of their heart are not working the way they're supposed to. So say we're not getting good strong signals out of our SA node, or it's not doing so at a good pace, someone may need to have a pacemaker installed on the outside of their heart to help them control that, okay? Now, one of the reasons that this might happen is because we might have developed other sets of nodal cells in the heart which try and take over. If those signals override or are too close to or around the SA node signals, it might be able to initiate a heartbeat that the SA node itself is not trying to initiate. Okay, so by installing another pacemaker, we can make sure that we're getting good, strong signals and that those signals are happening at the right time to cause the heart to beat when we need it to beat. Okay, like I said, that AV node is going to be around that 40 to 50 beats a minute, which is enough so you don't die, but it's not enough for you to do anything else. That means you're going to be weak and stuff like that and have dizziness, all that kind of fun stuff associated with it. But that's at least something that can be dealt with. The regular rate of other cardiac muscles is just simply not enough. So you have to at least have one of those nodal groups functioning. But if they start to go out, like I said, we can install pacemakers in folks, which is also pretty fun. Now, one other thing that cardiac muscle cells do differently, other than the fact that they're autorhythmic, which is cool, is that they contract very differently than a skeletal muscle cell. A skeletal muscle cell, remember, contracts in these little pulses, or the action potential wise, it does a pulse and then there's a break, like in kind of a resting phase, and then it can receive another pulse, right? And here, what we're going to see is we're going to depolarize, and instead of it going immediately back down to repolarize, instead, we're going to have a plateau, okay? Essentially, it's going to hold its contraction for just a moment, okay? This is once again going to make sure that we're able to empty the blood out. So these cells are not going to contract as fast as a cardiac muscle cell does, but instead is going to contract and hold, okay? And then release as it repolarizes. And this is basically going to allow us to make sure that we're getting the heart nice and empty before we repolarize. So because of this, these cardiac cells have a long refractory period. There's going to be a nice little break in between each contraction. They're not going to just you're not going to be able to pack electrical signals one on top of the other, one on top of the other, super, 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 super fast, the way you do for a skeletal muscle to make it hold itself. Like to continue holding tension on a skeletal muscle, you have to continually send an electrical signal to it. This whole time, I've never stopped sending electrical impulses to it, okay? So that's how we exhibit what we call tetany. That is not happening or not available to these cardiac muscle cells because of these kind of plateau natures of their contractions. Now, this gets us to our ECG. Because those muscle cells contract the way that they do with that little peak and that little plateau, we can actually monitor the electrical changes in the heart via all of the electrical signals that are being passed through all of those muscle cells. Because how do we send signals in a muscle cell? Electricity. Okay, so we can actually monitor those electrical changes. And this is what we're actually doing with that electrocardiogram. Now, electrocardiograms have three major pieces, or three major waves. Okay, the first one of these is the P wave. Okay, next we have what we call QRS complex. So we see a Q, the R, and the S. That's basically these 3 points here in this big kind of single waveform. And then finally, we have the T wave. So whenever the heart beats and you're, say, looking at an ECG or EKG, you're going to see this P, QRS, pause, T, P, QRS, T, P, QRS, T, and so on and so on and so on. 75 times a minute, okay? Or whatever their heart rate is, okay? Now, whenever you're looking at this, we do have some spaces in between these segments that are also going to be very important. We call these either segments, which are basically these lines here, or we can talk about the intervals, which are basically the spaces in between the starts and finishes of things, okay? And by looking at the space of something like our PQ segment here or our QT interval, which is basically our heart rate, we can look at this to make sure things are working the way that they're supposed to work. And if it doesn't look normal, that's what it means by not looking normal, is that, hey, this segment is too short, or this segment is too long, or our QT interval is too long or too short or something like that. And we can start to look at that and notice that we might have particular problems, because particular types of electrical changes in the heart will show themselves as particular changes in the electrocardiogram, which is why if you've ever seen a patient in, say, an ICU, they will have one of these on all the time. And you walk up to a nursing station and they're going to see their 2 or 3 patients, and they're going to have their 2 or 3 patients worth of EKGs just running so that they can look at them. And then if something starts to go bad, someone has a heart attack, someone falls asleep, you can tell just by looking at the EKG. And of course, all the other stuff that could happen in between something as ridiculous as falling asleep or having a heart attack. Now, what we're actually seeing then is the electrical changes in these 2 places. So the P wave going into the QRS complex, you're going to see this as the electrical changes of the atria, okay? Then the QRS complex, we have this really big spike here because of the big spike of electrical signal, because we have a whole bunch of muscle mass in the ventricles, right? Which means electrical changes going through. All those are going to be seen as a big electrical signal. And that's what's going to be our QRS complex. And then finally, we have another wave of changes, or another wave at the T wave, telling us what's happening in the ventricles. So let's look and see what's happening in each particular part. So the P wave, well, the P wave is the start of the signal, right? So that means our SA node has sent out its signal. So the P wave is going to be what's telling us to start atrial depolarizations. That means we're starting to contract in the atria, okay? Then we have a pause. Why do we have a pause? Because the atria are contracting. They are squeezing. The atria are being emptied, okay? Then we get to QRS. Well, what's happening at QRS? Well, that signal has now gone down here, and we've started to depolarize the ventricles, which means our ventricles are contracting. So then we're going to have this big pause in between our QRS complex and our T wave. Why? Because we need enough time for all of the ventricles to contract so that we can empty out all of the blood in the ventricles, right? If, say, our ST segment is too short and we don't actually squeeze this section all that long, we're not going to be able to get all of the blood out of the ventricles, which means the heart's not going to empty very well, which is a problem, right? That means we're not going to be getting enough blood to all of our tissues and stuff like that. So we could lead to things like lightheadedness and dizziness and all of that fun stuff. Then we also have this other little bump at the back, which is our ventricular repolarization, okay? So that means we're bringing, like, that's where that plateau falls off, and we repolarize the ventricles, and they're waiting for their next signal, okay? So that is our process. That's what's happening in each one of these phases. Once again, we've got some intervals to keep track of here. QT interval is essentially going to be what we're going to use to measure heart rate. If someone has a rapid heart rate, so this is what we call a tachyarrhythmia, that essentially means that QT interval is going to get real small, okay? Because it's just heart, like, beat on top of beat on top of beat on top of beat. Eventually, you can get so much tach that you don't get good spacing in between everything, and all of a sudden, heart's not emptying, heart's not filling, and you're not getting enough blood flow out to all these tissues and stuff like that, which is less than ideal. Let's put it that way. Eventually, you can also even lead into what we call fibrillation, at which point it's just chaoticness. So there's signals going everywhere, nothing's happening all at the same time. So blood isn't really flowing at all. And when blood's not flowing at all, that's a real problem. So if someone goes into VFib, they tend to do this, and then it just, not too long after that, it'll just be a straight line. Yep. And that's where all that fun stuff comes into play, okay? But once again, we can see all kinds of fun stuff in here. Like, say we have a block in our atrioventricular node, you might see this as an elongation of that PQRS interval and all kinds of fun stuff there. Essentially means that we have a slow conduction between the atria and ventricles, which means atria contract, we have a big pause before the ventricles contract, and it might be too much. So you're not getting enough blood going into those places. Because essentially the ventricles are not going to be able to get, or going to overfill, but then they're going to beat and they may not be getting enough signal. And this will tend to lead to some wonkiness down the road. Second degree means that atria just simply don't reach the ventricle. So it's really just the atria contracting and the ventricles aren't contracting at all, at least in the right pacing, which is real bad. So yeah, all kinds of fun things can arise and be seen via the PQRS and T waves. Okay, so our EKGs, which is why they are very, very useful and useful all the time. That's why we're going to do some looking at these. Now, to help us do that, I have a nice little machine right here. This, believe it or not, is a 6-lead EKG. Yeah. They actually have a 3-lead version of this that you can literally that's like a credit card. So for people that have very particular issues that need to monitor those things, they can. Yes. Those ones that I'm talking about there. I don't think these, but it is pretty entertaining. Until the crown on my Apple Watch broke and stopped reading it, I used to do the EKG thing all the time. Like I would just be bored, like, and be playing with it or trying to like get my kids to go to sleep. And it's like while they're trying to go to sleep, I was like playing with that. They are very fun. And of course, today of all days is the day I actually don't have my watch on, which is funny. All right, so the way we're going to do this is you're going to take our little thing here, and we've got the little arrow. You're going to point that arrow away from you. Okay, so the arrow has to point up, and the logo, so that V thing has to be pointing up. So you're going to point that arrow away from you, and then you're going to place it on your medial malleolus on your ankle. Okay, so you're just going to place it right there, and then you're going to put a finger from your left hand on one side, and then fingers from the right hand on the other. You're going to see this little thing turn green. If y'all want to walk over here, you can. See here. Okay, so it's going to turn green when everything's going correctly, and then EKG will read.
Oh.
Okay, so you're going to let it run for the 30 seconds that it needs to run, and this whole time it's going to be monitoring your EKG. And this is just the single lead version. All of the 6-lead stuff, like you can see stuff, but it's just for what we need in here or what we're talking about right now is unnecessary. We're just looking at a normal sinus rhythm, but look, we can see PQRST, PQRST, PQRST over and over and over again. And say normal sinus rhythm. Okay. And this we're going to kind of consider our normal resting rate. Okay. Then what we're going to do is after y'all have both tested this, we're going to do a little breathing exercise. And then we're going to do this again. And while we're doing a little breathing exercise, and we're going to see if we can get it to change. Then we're going to do a little bit of exercise and see if we can get it to change again.
All
Okay.
right.
Sound good?
Yeah.
Usually I get irregular rhythms, which is fun.
That does happen sometimes. So that might be one of the reasons to have one of those or have like a watch on that can monitor those things. No, my wife has had a couple of PVTs or issues with PVTs popping up. So and it'll work through your sock. Yep. Yep, turns green. Super clean. Jennifer, this is now be a great time to do the heart rate virtual lab or conduction system virtual lab. Although you can at least kind of see what's going on here on the visible body stuff. But I'm going to move this over to their table, maybe. So you can kind of see what's going on.
So we put safe and close.
Yep.
Safe and close.
Where did my Zoom thing go? Oh, there we go. Oh, it's the wrong angle. Put it on the other angle.
哦。
It'll stabilize. Just take this in. Not super strong. It's just a lower heart rate.
I used to, before I had surgery, I was at 54.
Yeah, that's pretty low. I would die. If my, yeah, that would be good. But your heart's also not having to try that hard. So that's also part of why your peaks aren't very strong. And that's also one of the things I want you all paying attention to. Because when we do this again here in a minute after exercise, you're going to see that the shapes of the peaks are going to change. Like the QRS will get stronger. The harder the heart's having to work, stronger that QRS is going to get.
Yep.
And the stronger the T wave you go. All right. So now what we're going to do is I want you to kind of sit down and close your eyes. And what we're going to do what's called a physiological sigh. Okay. We need to breathe in, or we're going to breathe in for about 2 seconds, and we're going to exhale for between 4 and 6. It needs to be at least double however long you need to breathe in. Now, if 2 seconds isn't enough time for you breathing in, you can either kind of pick up your pacing as you're breathing in, or just simply breathe in longer. But if you breathe in longer, you're gonna have to exhale longer. Okay. All right. So just kind of close your eyes, and we're gonna breathe in for 2 seconds. So. Breathe in. Breathe out. 6. Breathe in. Breathe out. 6. Just keep that in. Now, whoever wants to go first on this, go ahead and grab it. And just kind of keep doing this until you're kind of feeling good. So just kind of keep doing your breathing exercise. Can I have a picture? Yeah. So then whenever you're ready, just let me know. Yeah. So don't put your fingers on it yet until you truly do feel like you are relaxed and you've done this breathing thing enough. Okay. But then keep going. So breathe in. Breathe out. And just focus on those breaths. Breathe in. 1, 2. Breathe out. 2, 3, 4. Breathe in. Breathe out. In. We got your heart rate down to 60 at the end. Overall, it was saying 73. But we actually had it all the way down to 60 in that last little bit. So if we could have kept that going for another 10 more seconds, it probably would have had an average in the low 60s.
60,
okay. So this is a great exercise for if you notice that you are something's wigging you out, you're stressed or something like that, and you notice it everything's picking up or something like that. Doing that, just taking a couple of seconds. So that was less than a minute. So if you do that just for a couple of minutes, so 10 or 15 cycles, that's a good way of kind of getting yourself kind of recentered and balanced back down. This is actually why this is a very important tool for things like soldiers or people in stressful professions and stuff like that. Being able to just kind of get yourself okay, recentered and everything is fine. You can continue this over the span of for a few minutes and use this actually as a way to fall asleep even. Another way of doing it, so the way we're doing it now is 1 in, 2 to 3 out. That is a great kind of way of calming down. To go to sleep, the Navy SEALs teach what they call box breathing, or the same thing also to get them calmed down, where it's 2 seconds in. So they do a 2-second inhale, then they do 2 seconds in, then they do a 2-second hold, then they do a 2-second exhale, followed by another 2-second hold. Now, if you try that just for a couple of repetitions, you'll realize that to do this, you have to focus on it. Okay. This is not something that you can do while you're doing other stuff in your head. This forces you to focus just on this. And because of that, that also leads to the same thing. Heart rates can fall, and you can also use this as another trick to kind of, like, if you're laying in bed and your mind's going crazy, doing box breathing is a great way to kind of force yourself down. It forces you to kind of vagal down and fall asleep.
That's so cool.
So
Wow.
both of these are great little exercises that you can do. Either
Yeah.
1 in per 2 out, so 3 seconds in, 9 seconds out, 2 seconds in, 4 to 6 seconds out, or this kind of box breathing thing is a great way of kind of forcing you down.
Yeah.
Because it forces you to focus on this, because you can't really do either one while you're thinking about lunch or whatever. All right, so once again, what? Don't do away. No, come back.
Do I do which one?
It's gonna be your it seemed like your signal actually worked better on your right earlier. So go ahead and do your right, it's fine. So once again, do a few cycles of that breathing before you kind of put this on there. So just kind of get it ready. And then whenever you're ready, just tell me, and then yes. So whenever you're ready, just tell me and then we'll get it started. So just kind of do a couple cycles, say you're good to go, and then we'll get it recording. You can put your fingers on them because it's going to measure for 30 seconds. So yeah, just keep doing the breathing. And
Succeed.
Ooh.
so your signal's got to be pretty strong. So that is a great way of kind of getting ourselves to both chill out, but there's also one thing that we can do to help kind of mess with our rates. Now, go walk a few laps.
Around the thing?
Yeah, around the circle here. Not around the building, like, because this whole thing is just a block. So you can just kind of walk a few laps in here. And then we're going to check it while your heart rate's up. You're elevated with it. Either in the classroom or even outside the hallway. Wherever you want to go. If you want to be real crazy with it, you can start doing some jumping jacks, which is what I've done. What's crazy is it doesn't take long for this particular exercise to get your heart rate real high. Yeah. Because this is one of those ones that kind of hits everything. I did this for 5 minutes last semester. And when I stopped, I got my heart rate to 160. Although 5 minutes is a long time.
Yeah. Yes.
All right, I'll test mine then. All right, you can just keep kind of walking here just so you can see how ridiculous this is. See how tall the peaks are?
Yeah.
Here, you can go next. See, hers sped up quite a bit. Signals are stronger too. was gonna say, I thought I saw something.
I get weird, like little flutters whenever
Uh-huh.
I do.
Here.
Mm-hmm. It's cool to look at.
It's also weird.
Cause they thought about putting me
And
on a
that.
halter.
So
But
that's
no.
the flutters that you're talking
Yeah.
about. So that's a PVT right there.
Mhm. Oh, the terms. They're coming back.
Another little flutter.
Yeah.
That's cool though.
That is cool.
Wow.
They haven't told me like, I go in for like visits every few months
Mhm.
for it, but they haven't put me on like a halter or anything.
Yeah. All right.
I wanted to. My whole family has all these heart problems and they
Yeah, that might not be a bad idea then. Or at the very least get, like, if you have the means to get like something that you can kind of monitor yourself. My wife from home, like she doesn't like to wear a watch.
Yeah.
But she has an app on her phone that lets her monitor those things. Like where it can do it, it just doesn't do it quite as well as the sensors on the watch do.
Cool. No, I tried to use like one of the watches to do it, but I just had so many like external variations.
Yeah.
Couldn't.
No, there's lots and lots of good tools available now. So yeah, yours went up pretty good. Not a whole lot, but you also paused after a little bit.
Yeah.
But yeah, look at the peaks.
Mhm.
Like yours is beating harder than mine, but you're also young and an athlete. So
That's cool.
those days are behind me. Hey, I once ran a 57-second 400.
Really? Nice.
You didn't check?
Huh? For a little bit. No, they actually tested a bunch of us to see like a bunch of us that were set into the group of kids who were going to be like the distance runners, which is hilarious that I ended up in that group. I'm terrible. And like it was like, okay, we're gonna run a 400. We're gonna take a minute break, run a 400, take a minute break, run a 400, take a minute break, run a 400. And it's like, okay, add all those together, and that's your target mile time. My target mile time was like 4:30 something. It was like, no, that is no. And they're like, yeah, you're in the wrong group. You should go over there with the fast people. It was like I was significantly faster than everybody, like at each thing. But it's like putting it all together is like I just can't. Like I can't run that long. Like I actually did run a like sub 5-second 40. So I was like 4.9, 4.8. So I was reasonably fast. Like not like crazy fast, but I was fast.
That was your time for the mile?
Hmm?
That was your time.
That was supposed to be my target time for the mile. Like the fastest I ever ran a mile, like when I was in the fittest I ever like got, which was in undergrad. The fastest I ever ran a timed mile, I think, was 6 minutes and 10 seconds. Like I just don't have the endurance to go that long and that fast. Like go that fast for that long. Like I can run in a straight line for 10 or 20 seconds with anybody. But like it's when you have to run for multiple minutes, I just couldn't do it.
No, I'm very good at short distance. I'm more like a long distance.
Yeah, no, like I played baseball growing up, man. Like we ran 90 feet. Like very, very different. But yeah, so that's basically all the stuff that I wanted to talk about today. Those like getting the conduction system, the heart figured out, doing some dissections, how we regulate those rates, kind of what we're trying to do when we're regulating that stuff. That's basically the bulk of what I wanted us to talk about today. So have y'all got any questions for me? No? All right, so then we are good for today. That means we are done with module 1 stuff. So when we come back next week, it's going to be on module 2, and we're going to talk about blood vessels, which means next week when we come in, we're going to probably do a little bit more exercising and stuff to look at blood pressure. Okay, everybody good?
Yes.
Sweet. Then yeah, we're good to go.
Oh, I know.
There you go. See you Monday. Wow, my hair looks